right, well, thank you all for getting here early in the morning for our start on the second day, and thank you all for your participation yesterday. It was, I think, a great start to our inaugural event. I'm very pleased to welcome Eric Green, and I'll keep this really short because he's going to tell you a great story about his background and the history of genomics. But we're proud to have him as an alum of the MD-PhD program at WashU and former faculty member, and then has done some amazing work at NIH and NHGRI. So I'll let Eric take it away.
Thanks so much. Well, good morning, everybody. Thank you so much for the invitation to be here. I was delighted to participate in this. I've watched this entire experiment going on here in this part of the campus go on since its inception. I heard about it early on because I've always stayed connected to Washington University. And so I'm delighted to learn more, spend the day here, and start the morning by talking about one area of the biomedical enterprise that I'm sure many of you at least keep an eye on, or hopefully has been influencing what you care about, and that's this area of genomics. And so I'm going to tell you the story of human genomics, at least from my perspective. A story that has now gone on over 35 years. I've been a part of it from the very beginning. And so as a result, it's really interwoven with my life. It's also interwoven with Washington University. And I'm going to tell this journey really very much as a story. So first thing, I was born here, first of all. I mean, literally here, or at least 5-minute walk from here. And I'm a St. Louis kid. I was born and raised in St. Louis, West County. And I went away to the University of Wisconsin as an undergraduate to spend 4 years to get my undergraduate degree. But then I came back here as an MD PhD student, where I didn't work on anything related to DNA or genetics. Instead, I worked with Jacques Benziger and Irv Boehm, and I earned my PhD studying sugar molecules that get attached to proteins and help them do their functions as glycoproteins. And then I graduated in 1987. Now, this is a significant part of the story because 1987 is a very important year in the history of genomics, which I would categorize in some ways as, better lucky than smart. And the reason why it was better lucky than smart is, as all physician scientists, when they get trained, they face a lot of forks in the road of how much do I want to do research, how much do I want to do patient care, when do I want to get back to the lab, blah, 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 blah. That's the problem of being trained with having the flexibilities as a physician scientist. And I made the decision, and I don't think he's here, he was here last night, but partially influenced by Jeff Milbrandt, who's a prominent member of the WashU faculty, and a few others, that laboratory medicine was a great thing to go into as a physician scientist, in part because you got a foot in the clinical door, but you also got back to the laboratory very quickly. And so I became a resident in laboratory medicine here. And there I am. You can see what I look like. And then you immediately have to make this decision really within the first year, internship year, because by your second year, you go back into the laboratory. And so then I had to make the decision of I need to find a postdoctoral position in 1988. And I ended up deciding to work with somebody named Maynard Olson, who was this incredible intellectual giant. And I got involved in the Genome Project as part of he was one of the founding visionary leaders of conceptualizing the Human Genome Project for the entire world. And off I went running, and that's what I looked like during the Genome Project. And there are some of the earliest maps of the human genome that I constructed as part of the Human Genome Project. So that's my story of how and then I got recruited. I became an assistant professor here. Remember, Washington University got one of the first of 5 genome centers to participate in the Human Genome Project. And I was participating in that for a couple of years, but then I got recruited to the Genome Institute by Francis Collins, where I was for 31 years, and I eventually succeeded him as the director for 15 years. I'll have more to say about my circumstances at NIH near the end of my talk. So that's why I'm here. And that explains why I've been involved in this field of genomics from the beginning, and so why I think I'm a good storyteller for this enterprise. But I think you will also see how this story does have this arc of bringing me back to being a physician scientist. And that's why the title of my talk was From the Genome Project to the Realization of Genomic Medicine, because that's been my personal journey ever since I made that decision to go into lab medicine, get back to the lab, get involved in genomics, be part of the Human Genome Project, I never thought I would see genomics truly changing the practice of medicine in my lifetime, but we've done it. We've done it. So now let's go to the story of genomics. I would contend that two scientific fields launched last century are changing medicine this century. You may not realize, but let me just do a history lesson here. The word genetics got coined in 1907. It got put into the scientific literature the first time in 1907. Genetics is the study of heredity, of how traits are passed from one generation to the next, from one cell to the next. In 1907, when genetics was named, they had no idea even what the hereditary material was. But shortly thereafter, as the century progressed along, we figured out DNA was the molecule of heredity. And of course, many of you have either remember or you read about how the 1953 discovery of the structure of DNA was the final step and the piece of the puzzle of how it was that DNA could be that information molecule. And after the 1950s, everything took off in terms of understanding how DNA actually worked, how to manipulate it, how to clone, et cetera, such that it led to the naming of a new field in 1980s called genomics. The word did not exist until 1987. This is the first use of the word genomics in the scientific literature. Do notice that 1987 is the same year I graduated with my MD PhD, which meant never once in medical school or graduate school did I hear the word genomics, because the word didn't exist until. And of course, the whole idea of genomics was about being comprehensive, tackling an entire genome of an organism and studying it. The genome is all the DNA of an organism. It's the blueprint of life, if you will. And the reason all this became so exciting around the time I became a resident, in 1987 in particular, was because there was this incredible accelerated progress in understanding the structure and the function of DNA, starting with really the discovery of its structure. And that led, of course, to the elucidation of the genetic code, that classic lookup table we learned in like 10th grade biology class, that how it was that DNA encoded for amino acids. But then in the 1970s, we learned how to read out DNA, and by the 1980s, culminated in lots of technologies to do recombinant DNA work and PCR work and so on and so forth. And so as the 80s progressed, there got to be this drumbeat of discussion that, armed with this tool belt of basically technologies for reading DNA, for isolating DNA, for manipulating DNA, maybe they could all be scaled up and tackle genomes as an entire entity as opposed to just little bits of DNA here and here. And you started to see very influential meetings take place in the early 1980s that started talking about the idea of a big, audacious project. A very influential editorial linking the idea of, if we would understand the human genome completely, it would be critical for cancer. Absolutely was the case and continues to be today. The naming of the field in 1987, but then scientists got real serious, like Maynard Olson and many others, to start to convene major advisory groups that would put out reports, such as this one from the National Research Council, or this one from the Office of Science and Technology Policy coming out of the White House. And with all of those reports came the obvious idea of, we should get going and do something big, do international, but we're going to need money for it. If you're going to need money for it, got to go to the US Congress. They had their official hearings in 1989, which were the hearings that led to the funding of the Human Genome Project, and really off it went. Now, so 89, huge year to always remember in the history of humankind, because I would contend that really in 1989, things just changed forever in so many ways. First of all, Taylor Swift was born, and I think we all have to acknowledge that is game-changing in the history of humankind, okay? But at the same time, it was the year before the Human Genome Project was about to be born. And indeed, this will be my version of the Human Genome Project I'm about to tell you about. So that was 89. And there it was. I mean, I can tell you, right, I was just right there in the McDonald Sciences building, working with Maynard Olson and others, writing some of the earliest grants for the big grant for getting the first center here at Washington University. And off we went. We got the grant. We started mapping human chromosomes. The Human Genome Project began October 1 of 1990. Ended up taking 13 years. I'm going to now just fast forward and just say, wildly successful, accomplished all of its goals, ended early, and delivered the signature accomplishment of sequencing the human genome for the very first time. And the rest I will send you to lots of descriptions of the Genome Project because it really is in the history books now and gets talked a lot about as a really successful enterprise. In April of 2003, it was officially ended. Lots of celebration. And what we had, and let me remind you, what we had in 2003 was 92%, wasn't 100%, but 92% of the sequence of the 3 billion letters of the human genome. Of course, we didn't really put it in a book, but this is just a metaphor. You get the idea. There's a lot of letters. It was the order, but it was not the interpretation of the genome. It was just the first representative sequence of a human genome. Well, needless to say, this obviously set up a circumstance that, we had done it. We sort of had accomplished the first big thing in genomics. It was so unusual in so many ways. It was international, it was highly coordinated, it was top down, it was just not traditional science, but it had successfully accomplished all of its goals. So that was the finish line. But of course, it was also the starting line, right? When you think about it, just having a sequence of the human genome, that was just going to be the beginning of everything. And so, the finish line was just the start. And so, 2003 was a very pivotal year. And I will tell you that all the participants and all the funders of the Human Genome Project, they did go off in different directions. It wasn't just the NIH. There was other funders like the Department of Energy in the United States and other funders internationally. And they all were at the Genome Project for slightly different reasons. Why was the NIH there? Well, it was the recognition that if we really were going to figure out how to better practice medicine, we needed to have the tools to understand the unique blueprint of each of our patients. Because I can tell you, I got a great medical education here at WashU, but everything I was taught in medical school was about the average patient, because we didn't have the means by which to be able to decide differences among patients, at least with respect to their blueprint. That's at least their genetic information. And that was clear that was going to be enhanced if we could have the tools for being able to understand each patient's unique genetic blueprint. So the idea, the pivot that had to take place at the National Institutes of Health, particularly the National Human Genome Research Institute, was to think about, now that we have the first sequence of the human genome, how are we going to actually operationalize it to eventually change the practice of medicine? Well, it should come as no surprise if that was the rationale for the Genome Project, at least from a health perspective, that the popular press or the scientific press was going to do the exact right thing. They were going to juxtapose the two obvious words, genomic and medicine, and put them together. So sure enough, shortly after the Genome Project ended, everybody was talking about genomic medicine. And it was very exciting. It was clearly going to be what we wanted to do. There was a slight problem with it because in 2003, we could spell genomic medicine, but it was a very blurry concept. How are we ever going to get to the point of getting unique genomic information about each patient and do that to help manage their clinical care? Well, you had to start by understanding what it meant. And what genomic medicine is using genomic information about an individual as part of their clinical care. It's not to say that's the only thing relevant. Of course, that's only one component of human health. Obviously, physical and social environment is important, but genomics was going to have a laser focus on trying to understand the genomic information that's unique, that each of us carry with us into every single clinical encounter. And so there was going to be this pretty important pivot. And our institute did it on behalf of NIH, and eventually we brought the rest of NIH along with us. And the idea was a new starting line, the end of the Human Genome Project, and a new finish line. The finish line was going to be far more than 13 years. It was going to be more vague. It couldn't be articulate. It was just going to be realizing genomic medicine writ large. And we also realized that this was going to be the second big journey in genomics. First one was the Genome Project. And just like any journey, we knew we had a lot of steps to go, and we didn't even know what all those steps were. Same with the Genome Project. Lots of steps, a lot of uncertainty, needing to be nimble along the way. But what we realized in the Genome Project, you just got to take one step at a time. And then when you finish that step, you celebrate, and you check it off your list, and then you get back and you figure out what the next step is. And you just have to keep iterating this, and slowly but surely, you'll get closer and closer to your finish line. I think we also realized that even more than the Human Genome Project, this journey was going to be more complicated. It was going to take longer. It was going to involve more disciplines, and it was going to, of course, be international, and we had to expect that it was going to be more akin to a very long marathon, or maybe even a super marathon, and we got to go in for the long haul. Now, a very reasonable question to ask is, okay, how is it that, that genomics, how do you figure out how to make this progression to get these journeys done? And that's what the National Human Genome Research Institute was responsible to do at NIH. And when we thought about, when the Genome Project ended, how are we going to do that, we decided we would continue the playbook that worked so well during the Genome Project. And the playbook was, don't assume you know what to do as a funding agency. Rather, use your community to help figure out what you need to do, listen to them, engage them, and then try to synthesize it and write it down. And there were 3 very influential, successive descriptions of the strategies that were going to be needed to get us through the Human Genome Project that's shown on this slide. And so NHGRI decided to continue to do the same thing. Most other institutes don't do it this way, but we thought it worked well for the Genome Project. And so over the years, starting the day the Human Genome Project ended, we put out a strategic vision, which represented the culmination of a 2-year strategic planning process that involved engaging hundreds, probably thousands of scientists around the world to say what's next. And then you realize that these things only have a shelf life of about 8 to 10 years. I became director in 2009, and so I was responsible for overseeing this strategic vision, and the most recent one came out in 2020. So I will tell you that in the last 22 years, these are the 3 strategic guides to what has happened in human genomics. And you will see that each of them sort of gets a little more clinical along the way. And I don't have time to go through any of them in any detail. But what I will do is just give you a brief highlight of what I think are the major steps that were predicted in those strategic visions and then have been executed either completely or, as I will tell you, partially along the way. So as we've witnessed progress over the last 22 years to this journey from the Genome Project to the realization of genomic medicine, what have been the major steps and how far have we come? Well, I can tell you that the day the Genome Project ended, it was very clear what that first step needed to be. We had just finished sequencing the human genome for the very first time, and it cost about $1 billion. It was well worth it, but it was a little expensive for a clinical test of an individual patient. We realized it. Literally, the day the Genome Project ended, we claimed and wrote in our strategic vision, we need a $1,000 genome. You may have heard that phraseology, the $1,000 genome. I'll take a little credit for that sentence because I was the clinical pathologist writing along with Francis Collins, the other leaders of the Institute. I said, look, I understand what a clinical test should cost. $1,000 is nice, rounded, and it's a reasonable cost for a comprehensive test like that. And off we went. NHGRI gave out lots of grants. The private sector got involved. Venture capital got involved. There was a lot of companies. And to tell you what, it took a number of years, but we did it. We reduced the cost of sequencing a human genome by more than 1 million-fold. And that was, just absolutely game-changing. We were allowed to sort of cross that off our list. How did we get that done? Well, all new technologies came on board to replace what was the $1 billion genome, which were factories like this, and there was one just down the street here at WashU that looked just like this, with these very expensive machines running 724, lots of people. That's what it took to get that first genome sequence. But the $1,000 genome was going to be all new science fiction kind of technologies. Some of you in the room know what these are, and there's multiple ones, and they keep coming. In fact, now we talk about $100 genome, and whether it's Illumina or PacBio or Oxford Nanopore, whatever, you could read about it. We tracked it at NHGRI, and you just saw this precipitous decline that by about the year 2018, 2019, we went below the $1,000 threshold. And so being able to cross that off our list was, and the rest, and by the way, that has changed everything, not just for human genomics, but across all of life sciences, because when you have cheap DNA sequencing, a lot gets done. Well, you empower a bunch of scientists and clinicians with the ability to sequence genomes for less than $1,000. You're not going to just hang out and just look at that first human genome sequence. You're going to want to sequence lots of genomes to get at how are we different, because that's the most interesting part of genomics, is how people differ. And we've now sequenced millions of genomes, and we've been able to cross that off our list. I mean, many millions of genomes at that point. And what we really are trying to get at in doing this is that we need catalogs of our differences. Some of us have very unique differences, but most of our spelling differences on our DNA are actually quite common, and we should be able to have catalogs of those so we can figure out what they mean. For those of you who don't live in a human genomics world on a regular basis, if you take any two people, We are about 99.6% identical, means we're about 0.4% different. But that means that every one of us carries with us this big universe of 3 to 5 million spelling differences between any other person in this audience, most of which have no consequence on our biology, our physiology, or certainly our health. But we got to get at the idea of what are the smaller subset that are biologically relevant, and then an even smaller subset that are medically actionable, that are relevant for clinicians to be aware of. And of course, that is a Herculean task that we're in the middle of. And what's critically important is that when you see a DNA spelling difference, to be able to know whether or not it's going to be influential biologically or medically, you need to understand how the human genome actually works, so that you know whether or not a spelling difference influences it. And so here was the third step that we needed to be working on, which is, of course, understanding how the human genome sequence actually works. And I think we've made, in the first 22 years, profound advances in understanding how the human genome functions. But I will tell you, it's a complicated 3 billion letters. It's a really complicated language, and we've just used 22 years to cream skim the best we can on what we understand. But I tell my adult kids, your generation is going to be interpreting the genome, and your kids' kids, and so you could tell any of your grandchildren, they'll be interpreting the genome sequence, because I think it's going to be a multi-generational challenge. Now, I will give it, therefore, a medium check mark, because we've got a long way to go, but we've made incredible progress. And in fact, the progress has gotten so good that you couldn't even keep up with it unless you have tools to be able to teach you how to do it. And of course, we've made all those tools available, like everything else in genomics, freely available to everybody with an internet connection. So for those of you who don't know, you can go onto any browser, or you can jump on an internet connection, go to browsers that look like this, which are just basically layers and layers and layers of functional information about the human genome, like where are the genes, where are the regulatory elements, which parts of DNA gets made into RNA, where are conserved sequences, blah, 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 transcription factor binding sites, a lot of biology there. Don't worry about the details. Just think about the concept that what we are doing now in genomics is making a Google map of the human genome. Instead of where are the hotels, where are the restaurants, where are the gas stations, we're just saying where are the genes, and where are the regulatory elements, and where are the other important functional elements. And so because of that, we've made significant strides in understanding how the human genome works. But I'm realistic. What is known is a very small subset of what we still don't know. And all the efforts going on this campus and every other life science campus around the world is just working hard to push that iceberg, more and more of it, above the surface, so that we have more that is known. Because there's still a lot to be learned about how the human genome works. It's just the tip of the iceberg that we know about. But we don't even know what most of the 20,000 genes do. We only know about a small subset. There's a whole epigenomics landscape where the DNA is getting decorated and being influenced by environmental factors. We got to understand that language. RNA is a very complicated molecule. It's not as simple as we once thought, certainly not what I was taught when I was in school. There's a lot of biology in RNAs, and we're learning those as well. A lot of choreography going on turning our modest number of genes on or off in different cells in different times under different conditions. And then there's, of course, proteomics and glycomics and metabolomics and a lot of other physiology relevant big data omics type fields. And all of these are areas that are currently being tackled. But these first three steps still seem far away from human health. And so, but as we got better ways of sequencing DNA, better understanding of human genomic variation, better understanding about how the human genome works, we obviously could turn our attention to saying which of those variants influencing which of those functional elements in the genome are conferring risks for getting human disease. And I would contend that we have made significant advances on unraveling the genomic bases of human disease. Now, once again, I'm going to have to only give this a medium check mark because the story I'm about to tell you is progress in some areas and less progress in other areas, but we still should recognize the first 22 years of having our blueprint in front of us, we've made a lot of progress, and in particular in rare genetic diseases. So let me remind you, all diseases, essentially, all diseases have a genetic and genomic influence. They vary by that influence, and they vary whether or not they tend to be rare or they tend to be more common, although there's a lot of blurring between them, as I will admit to in a few slides. But rare genetic diseases end up situations where when you have a disease and you end up either with a single mutation in that gene if it's a dominant, or 2 mutations if it's recessive, shown here as a dominant disorder, you have significant risk, almost nearly 100%, if not 100%, but sometimes just short of 100%. And that is the circumstance of what you are looking for. And another way to think about this is, yes, there might be a small environmental influence and a couple of other variants that influence the disease, but by and large, the major risk factor, somewhere significant, sometimes approaching 100%, will be due to one gene that you are searching for. And so the search is a bit more straightforward. Here we've made remarkable progress, and there's a lot of celebration that is well deserved, and I'd like to put numbers on things when I can. The curtain went up on the Human Genome Project in 1990. There's an estimated 10,000 rare diseases. That number is still being precisely defined, but something on the order. In 1990, when the Genome Project started, there were only 61 rare diseases for which we knew what the gene was. One was sickle cell, Lesch-9 syndrome. Cystic fibrosis just got in under the wire in 1989, but only 61 of 10,000. Let's fast forward to today, empowered with all this incredible progress in genomics, and it's impressive. We're now over 6,000. We have made this incredible progression from 61 to over 6,000 rare diseases for which we know the exact gene and the underlying genomic basis for it. Now, we still got ways to go to get to 10,000, but this has been game-changing in so many ways. It's taught us a lot about physiology, but it's also really helped a tremendous number of patients and have led to some cases therapeutic opportunities and so on and so forth. This is a success story. And this is the challenge, because if I walked over to Barnes, BJC, whatever it is these days, if I did rounds, or I walked in any of those clinics, most of the patients there aren't there because of rare diseases. Most of them are there because of common diseases, where there's typically thought to be multiple genes involved, and typically the risk, it's multiple genes, multiple variants that are conferring little bits of risk, but not 100%. And so it becomes a much, much more complicated circumstance with what is also a greater influence of the physical and social environment. And so teasing out those small contributions turns out to be really complicated and, to some extent, a good scientific debate. Many of you who follow this, some of you, there's this whole era that we've lived through called genome-wide association studies, where we have basically peppered across the human genome regions that we know have variants that are statistically associated with risk for getting a disease like hypertension or diabetes or cardiovascular disease and so forth. It's not causation, and it's only one small piece of the puzzle. And that gives us lots of clues to where to look. But it doesn't give you the answer, it doesn't give you the gene, and it still requires a lot of work. Some people believe this has been a fool's errand, that it hasn't really revealed enough, and others believe this still is the way to go, and it's just a matter of enough time before we figure all this out. But we have hundreds of thousands of these statistical associations with regions of the genome that are associated with a disorder we're interested in, a health trait we're interested in, but we don't yet have the answers. And so, in aggregate, rare diseases, as I told you, really good success. Common diseases, a long way to go, and even some debate about exactly what is the underlying architecture, and is it how common are these variants, how rare are these variants, and so forth. And then, of course, life's always more complicated because this is not a perfect dichotomy. There's a lot of diseases that sort of feel a little bit like a rare genetic disease, and another one that feels a little more. So there's a lot of blurriness. Biology is always complicated, and but I think we've made good progress. But obviously, we have a long way to go in fully understanding this. Well, this is great. We've gotten to the point of having translational research going on to giving us clues about the genomic contributions to rare and common diseases. It still doesn't mean that's medicine. That's just still sort of just shy of actually interacting with the patient. But I can report that we now do have vivid examples of genomic medicine that are emerging. I'm going to contend it's a small check mark because I'm so optimistic that, once again, it's just the tip of the iceberg, that the earliest examples I'm going to tell you about are just what we're going to see in much greater form as we get more and more innovation and implementation in this space. But still, as a physician scientist, I never thought I'd even see these examples in my lifetime, let alone on my watch. So I'm particularly proud of this. Now, how did we get to this point? We got to this point because we got cheap enough sequencing that we literally could sequence a patient's genome and we could compare it to a reference, available genome sequence, and we could figure out make a list of those genomic variants, 3 to 5 million of them, and off we could go implementing genomic medicine. I'll come back to this slide because it's an oversimplification. I'll come back to it later, but this is the basic idea. We got to the point where we could do this and start to explore it in different areas, in different ways of medicine, and actually begin to actually see changing the practice of medicine through the use of genomics. So what do I think are sort of these early examples that make me so excited? I'm just going to touch like 5 of them because it just illustrates that genomic medicine is real, even though it's still early days. The first one is cancer genomics. I already know there's people in this room that are far greater experts in cancer genomics than me. So I will just touch it by reminding you that cancer is a disease of the genome. The reason cells grow out of control to become tumors is because their genomes have picked up a bunch of mutations that make them grow out of control. But we can open up a tumor and read its DNA just as easy as we can open up a normal cell and read its DNA. And boy, have we done that a lot. In fact, one of the earliest examples of actually doing that took place on this campus by Tim Lay and Rick Wilson and Elaine Martis and others. And that was I guess now it would be 17 years ago. But in 2023, they had this article that celebrated that 2008 publication that they had sequenced a cancer genome for the first time and learned a tremendous amount. And I will just summarize a huge body of work. In fact, it's a whole new field. Cancer genomics is a whole new field, way beyond me. In fact, the National Cancer Institute has sort of taken it over as they should, rather than the Genome Institute. But this quote just says it all out of this article. Today, genomic sequencing as part of clinical care has transformed cancer diagnostics, clinical trials, and the use of new therapies to improve outcomes for people with cancer. Our unprecedented view of the cancer genome empowers clinicians, computational biologists, and bench scientists alike to define biologically relevant groups of people with cancer, direct genomic inquiry, and ultimately identify new therapies and biomarkers. It's just been game-changing. There's companies coming up around here, right here in this area, related to this. And I actually am very clear about one thing. I think 30, 40 years from now, we'll look back. I think the greatest impact of genomics on medicine will be in the cancer arena, because I just think they're just so obvious how they're so intertwined. So cancer genomics, a field in and of itself, already examples, vivid examples of saving lives, developing new therapies, better diagnostics, and so forth. But again, just the tip of the iceberg. Second area, I sort of foreshadowed. If you have knowledge of 6,000 rare genetic diseases, this starts to become diagnostic opportunities, and boy, has it been. Genome sequencing is now a standard diagnostic tool in medicine. You encounter a patient, especially a young child, developmental delay, a heart abnormality, cognitive delay, et cetera. Yes, you could start to do your standard diagnostic workup, but heck, for $1,000, sequence that child's genome. And in fact, this goes on multiple times. You're going to get answers, not always, but quite a few times you'll get answers. And what's really important is that it can mean diagnostic odysseys end much earlier. Numbers, about 40, it is estimated, and I think these numbers are hard to estimate because the curve is going up so quickly. Something like 40 to 50,000 individuals will get their genome sequenced just here in the United States alone, probably double internationally this year, as part of a workup for a rare genetic disease. And the fact of the matter is there's lots of good review articles out there. You could read about it. It's not that you get an answer all the time, but the latest reviews are sort of saying that it yields a diagnosis for a rare genetic disease about half the time. And this number will get better, because this is the floor, certainly not the ceiling, because there's so much of the human genome we still don't understand functionally, so that's like a black hole to diagnostics. And then there's so many examples where you have a variant and you don't really know how it influences, because we haven't gotten good enough at always going from a variant to functional consequences, so we'll get better at that. And trust me, AI and all the obvious things are going to help us do that. A lot of money is being invested in this as well. And in fact, I just happened to see this article, just came out like 2 weeks ago, so I threw it in, where there's now, the implementation is full bore around the world in terms of genome sequencing as a mainstream technology for working at patients with rare diseases. There's also some fascinating niches to follow in this area as well. One is just, I gave you the example of a child with heart disease, but there's niches like in neonatal intensive care units, where this has been really seismic, where you have patients in a NICU and they're acutely ill. Nobody knows what's wrong with them. If you can get an answer quick, you can save their lives. So starting in about 2014, NHGRI actually funded an investigator to develop a rapid genome sequencing approach that has now taken off around the world. And that was in the NICU, and then they started to do it in pediatric intensive care units. And there were so much uptake and so many studies that were done that by 2024, they actually did a whole review on this. And again, I only have time to give you a quote, but this was a sort of a comparison of the US and the UK, and is 44 studies already of children in ICUs with diseases of unknown etiology. 37% received a genetic diagnosis. 26% had consequent changes in management, and net healthcare costs were reduced by $14,000 per child. In the 5 years, there's a potential for infant and childhood mortality in the US and UK to have been reduced by several percent through the use of rapid genome sequencing as a first-tier standard of care test for children in ICUs with disease of uncertain etiology. And remember, the bill in an ICU is like $10,000 a day. The idea of getting a genome sequence for $1,000, or even if you have to rush it for $2,000, it's just cheap. I know, I was just in a meeting earlier this week, some NICUs are going to start doing every baby that gets admitted to a neonatal intensive care unit will get a rapid genome sequence. I think this is going to become standard around the world, hopefully, or certainly around the country in a matter of the next few years. And I thought this was really cool because I often have wondered if it's working in a pediatric ICU, what about adults? If you're an adult and you're otherwise healthy and you find yourself in an ICU and it wasn't because of trauma or because of poison or because of an accident, something, what is it? Is it maybe that there's an underlying condition that sure enough, they did this at University of Pennsylvania. Make a long story short, they looked at a bunch of patients, excluded those with obvious things like trauma or known disease like cancer, and then sequenced their genomes. And in 25% of, it was that big part, 25% of those patients had an underlying rare genetic disease that was unknown. Again, it's just fascinating to think about ways of stratifying patients in settings, especially expensive settings like intensive care units. Something to think about. Third area actually ends up being the most used area of genomic medicine in terms of total test number. It turns out that for years before genomics, many parents chose to learn a little bit about genetics of their unborn child, looking particularly for aneuploidy. That's like 3 copies of 21, Down syndrome as it's known. But that would involve an invasive procedure like an amniocentesis. Yeah, we don't need to do it that way anymore, because it turns out that the placenta naturally sheds small amounts of DNA, which is fetal DNA, into the maternal bloodstream. And using these way cool technologies of sequencing DNA, you can end up basically taking a blood sample, taking the cell-free DNA, which is a mixture of maternal DNA, but also baby DNA, and just counting sequence reads, counting how many times you see bits and pieces of DNA. And if you have too many from any one chromosome, like here on chromosome 21, you've now figured it out. So, the baby and the mom are releasing DNA, but now we can, as a screen, a first-line screen, just do a blood draw. It's non-invasive. It doesn't risk the viability of the pregnancy, which amnio can. It's much more pleasant than an amnio. And as a result, this has become the #1 genomic test worldwide, in part because insurance companies pay for this. And it is estimated that 7 to 8 million pregnant individuals will have a non-invasive prenatal test done this year alone. And it has just taken off and is now standard of care. The next area is an interesting area that relates to the fact that we all respond to medications very differently. And in fact, the track record's not so great. I mean, 10 years ago even was this article in Nature that talked about these 10 very commonly prescribed medications where for every person in blue where the individual responded well, were all the people in red where the individual didn't respond well. And the truth is, some of the differences, or a good part of the differences in our response, relate to variants in genes that are part of pathways of drug metabolism. And so the whole idea is that it's a very imprecise way to practice medicine. Could we do better? Could we find the right drug, not by inventing the right drug for each person, but you have 10 hypertensive medications, 10 asthma medications. What's the right one for that person based on their genomics? So the basic rationale of pharmacogenomics is stratify patients. And by doing take all the people that are mixed in the middle and do a DNA test, and we can get a DNA signature that find out which of the patients where the drug is not toxic and it's actually beneficial. And if we could do that, we can get to the point that we will no longer have sort of one size fits all for medications. We stratify these people and we pick the right drug, but also sometimes could even pick the right dose based on that, and off we could go. And is this realistic? Well, these data are hard to get, but for complicated reasons. But of all the FDA-approved medications, 14% of them are known to have a gene that influences their metabolism, for which it has been found that there are variants that influence whether or not that's an appropriate drug for you to get. And those drugs, which constitute 14% of FDA-approved medications in 22, are 10% of all medications that are given out, all prescriptions in America. These numbers are probably higher now, and they're only going to go up and up and up as we learn more and more about the genomic basis of drug response. But this is real. And companies, I'm not endorsing Invitae, but companies offer panels of these variants that are these genes that you could test for variants. And we're not talking about rare diseases these are commonly prescribed medications that are used to treat conditions that these medications are frequently used. It turns out that uptake has been slow, and we could spend hours talking about this, and we have many studies out there trying to study why do physicians not want to utilize pharmacogenomics. So a lot of this is an implementation problem and a little bit of insurance company problem. I think it'll get better. So I think right now there's some great examples, and at some medical centers and some health systems this is being used widespread. And around the world there's some places where it's being used much more aggressively, but there's just been an uptake problem in the US. Some of this is sociologic and some of it is financial, but I think it's something to keep an eye on going forward. And my last example really is one of I don't want you to think that everything about genomics is just about the practicing medicine once you have somebody who's sick. There are good examples of prevention that are worth noting, particularly for rare diseases, because rare diseases, we now have a lot of experience where if an individual like this gentleman, if he has colon cancer and then you find out his sister has colon cancer, they'll test the whole family. That's called cascade testing. They'll test the whole family for known genes that have mutations, and you could end up very effectively helping that family to find out who has the mutations that'll make them at greater risk for colon cancer and just get them screened more. Same thing for breast cancer, same thing for a number of things. Similarly, you could have lots of things in the pediatric stage with some of this we do at birth, some comes on later, where you end up in circumstances where you know that diet changes could help slow down cognitive delay or prevent cognitive delay. And if you could just get that to that early enough, you do that before there's damage that starts to happen. There are increasing numbers of opportunities for prevention for rare diseases, and as a result, it's just here and now, and it's very, very, very effective. There's a lot of discussion about the concept of polygenic risk scores, which is using data coming out of these genome-wide association studies to stratify people without knowing anything about the biology of the disease, but just to be able to place people at higher or lower risks. I don't have time to describe this in detail. I'm happy to talk about it at the break. This is an area of significant research, significant excitement, and occasionally significant concerns, because some companies are starting to just sort of offer this without having all the clinical data behind it to say that it's valid. It is an area of research, is what I would say. Maybe we'll get to a tipping point, and some clinical labs are starting to offer it, but just keep an eye on this. It's exciting, and I'm open-minded to it, but it is one of these things I still think it's a research area, not a clinical implementation area. So what I've just told you about is a 23-year romp through where we've gone from the vaguest idea of what genomic medicine was going to be about, and now I can show 5 major areas where genomic medicine is really beginning to make some contributions. If nothing else, it has given us clarity on what genomic medicine looks like, even though it's the earliest examples, but it's helped us solve a lot of problems around implementation that I think will make some of these other areas easier, or at least we know what kind of questions to ask. And as a result of that, those of us in the field, we're constantly looking at what's the next thing to come? What's the next thing to anticipate based on these earliest successes? So a lot of times I'll get asked, like, what's on your watch list, Eric? What do you think? Well, my watch list, I think I have to put in, there's a lot of excitement about this and a lot of discussion you're going to start to see about newborn genome sequencing, not ill newborns, all newborns. And this is being led for a variety of reasons. One, it just makes a lot of sense, but there's a lot of good research studies that are coming out, and I would just point you to a whole growing body of work where they are doing studies where they sequence a whole lot of babies' genomes and see what they learn and how it affects clinical management of these babies, especially as they grow up. It's also led to the creation of a new consortium that's actually meeting next month in London. I went to their meeting in New York last year. And this is called the International Consortium on Newborn Sequencing. And each of these little, whatever those little things are that point to a part of the world map, in every one of those places are population scale, or in other words, large scale studies of newborn genome sequencing of healthy newborns. So worldwide, this is getting a significant amount of attention. Examples of these studies to keep an eye on is the Guardian study. The Genomics England study is the one at the top, where they're taking 100,000 newborns in England are going to be studied. And here in the United States is the Guardian study that's going on. And so again, these are big studies that are going to give us some insights. Just two examples from the previous map. And then if we're going to keep up with the UK, this was breaking news from a few weeks ago. The UK has just sort of declared that within 10 years, they're going to sequence every baby's genome at birth and put that into their medical record. And now they have a single payer, so I know it's a different medical system, but it's quite interesting to see how internationally, especially the UK, they're really pushing the envelope on newborn sequencing. I think it's a fascinating thing to follow. So let me celebrate and congratulate this remarkable field. I do think we deserve a bunch of champagne bottles. But I do want to point out and just spend about 4 minutes saying that sometimes when I get to this point in the talk, I'm a little enthusiastic, and I know I am, and I'm very proud. But I also am realistic. This has not been a straight shot over 35 years. It was a lot of twists and turns and a lot of headaches and a lot of challenges, and we have more challenges ahead. So I want to temper my enthusiasm by also telling you that those who are involved in this field, we worry a lot. That's our job. We worry, we face the challenges, we think about what are going to be the obstacles. I could give you a whole hour of all the challenges, and there's a lot of them. But just as a highlight, I'll give you two. I don't want you to leave my talk thinking that we can just sequence a genome and understand everything immediately. This was an intentional and profound oversimplification. I guarantee you, I could sequence a genome of any patient at Barnes Hospital. We could get it done in a day or two. It'll cost $1,000 or so. I can get the best minds at WashU, best clinical minds together. We could round on that patient all together. We'll see the list of 3 to 5 million differences in their genome, and most of those differences, we will have no idea what they mean. We are just skimming the cream on understanding what we understand, but most of what those differences are, we don't yet understand. It is a huge area of research. I just sort of remind you what that research involves is taking every patient and their 3 to 5 million variants and then filtering and sorting and analysis and all sorts of things using databases and data resources and knowledge bases and getting it down to maybe 10 or 20 variants and then trying to say what you think might be important about those variants. This is a massive ongoing research effort. I can tell you the Genome Institute invests considerably into this process. We're getting better every month, every year, but it is not push button. We hopefully will get that way. And we certainly will need to get that way if we're to the point of sequencing every newborn's genome at birth. A second area of challenge is just, we live in this complicated world, and its society brings in a lot of circumstances that are quite challenging. Maybe the simplest way I could say, 'cause again, when you think about this 37-year arc I've been involved in this, when I joined, as a postdoc, Maynard Olson's lab, and even when I was working on the Genome Project, it felt like we were kids at that kid table at Thanksgiving back then. I mean, we were nerds. We just talked about mapping and sequencing the human genome. Nobody was paying attention to us for the most part. We didn't have any real problems except just getting the job done. But boy, has life changed for us, because now, no, Eric, you can't sit at the kids' table. I don't care that they're talking about iPhones, Instagram, and Taylor Swift. You have to stay here and talk about mortgages, life insurance, taxes, politics, and genomic medicine and healthcare. What do I mean by that? Well, all of a sudden, genomics touches healthcare now. All of a sudden, genomics is relevant in society, and we got adult problems. And we're dealing with these. We have an entire program that's been going on since the Genome Project began, studying the ethical, legal, and social implications of genomic advances. That's the ELSI research program. And we deal with real problems. Before, it was about patenting genes and genomic issues about whose genome are you going to sequence in the Human Genome Project. But now in healthcare, you take on all those problems that healthcare always has, especially in this crazy country of the United States where our healthcare system is so incredibly complicated. And you got to deal with how the healthcare gets delivered, how that healthcare gets paid for. You have to deal with all the regulatory oversight, responsibly so, because genomics is now a diagnostic tool. You got to worry about health equity. You got to think about the consequences of genomic technologies exacerbating health disparities. like all health information, you got to think about genomic privacy. And we have to remember, this is happening fast and furious. My medical classmates, most of which are still practicing, never heard the word genomics once, but they're out there on the front line of practicing medicine when some patients are coming into their offices with their genomes already sequenced. And then we have to think about the next generation that we're training right now, not just of physicians, but nurses and pharmacy students and nursing students and physician's assistants. Oh, and by the way, let's think about the patients. They got to understand some fundamentals about genomics if they're going to have an informed conversation with their healthcare professionals. So literacy is a big deal. And so bottom line is there's these challenges and there's many, many more. And that's all part of the responsibility of seeing this journey take place. You got to take the scientific advances, but you also have to responsibly think about what the societal implications are as well. So this 35-year journey really started knowing what the double helical structure was of DNA, using the Genome Project to magnify it. But ever since the Genome Project ended, the threads of genomics just continually get woven into this growing, beautiful tapestry of opportunities in human genetics and basic science and physiology, increasingly clinical care. And it continues because we're still actively stitching this together and making genomic medicine a reality. So that's the story of human genomics. In the last 2 minutes, I do have to acknowledge I'm standing before you. And there is an elephant in this room. Some of you may have noticed that on that very first slide, it didn't say director of the National Human Genome Research Institute. Even my badge says National Human Genome Research Institute. It said former director, because I'm not there anymore. To make a long story short, I drew the short straw, and there's 27 institute directors at the NIH, 27 institutes, 27 institute directors. We work directly with the NIH director, and each of us serve 5-year terms. Those are very official terms, and you can be reappointed multiple times. I drew the short straw that my reappointment date was the first one in the new Trump administration, and they came in wanting to decapitate NIH leadership and do a lot of damage, which they've successfully done. And my date also happened before the new NIH director even arrived, and they just chose not to, even though everything was set, they just chose not to reappoint me, and so I was forced to retire. And so, as I like to joke, I failed to dodge the bullet. And some of you may have seen this. It did hit the news. I'm no longer I'm no longer there. I'm now the former director, and I'm sad about that, and my staff is sad about that. But I go on. I'm very optimistic. I always like milestones, so I'm not going to dampen my enthusiasm for my run at NHGRI. I was incredibly proud to be the first NIH director appointed under this president who liked to smile. And now, this president, he doesn't like to smile in his official portrait. I was actually the first institute director fired. And there's now been 7 of us, of the 27, who I wasn't really fired. I was just forced to retire, but that's why I put the fired in quotes. But I will say, everybody's been so wonderfully supportive. I've gotten lots of notes, including from the leadership of WashU, who were very sorry to see me, always been very proud of me, and were very sorry to see me lose my position at NIH. And so I got lots of good emails, lots of good notes. My favorite was a handwritten card I got from a colleague. I won't say who it was, but this was the cover of the card, and I just thought that was really good. Thanks for not being a conspiracy-addled, science-denying idiot. I took that as a compliment. But the bottom line is I'm not going to let it dampen what I have seen as this incredible progression where we went from such a small community involved in the Human Genome Project at places like this and a few others. But over the 35 years of my involvement and many thousands of others around the world, we've seen building up this incredible community of people that have now gotten it to the point where we're seeing genomics truly improve the practice of medicine, and by everybody working together, standing on each other's shoulders, and it's still going. And it's going to be great to watch this, and I'm not going anywhere. I've retired from federal service, but I have not retired from being an active member of the genomics community. I will continue to be involved in many, many different ways. And so with that, I will stop, and whatever time is left, I'm happy to take any questions. And I'm here all day, or I'm here all day, but many of you are here in the morning. Catch me at the break. Happy to talk about anything. Thanks very much. Questions?
There we go.
Yeah,
for a few.
I see one back there. Yeah.
First of all, great talk and great body of work. But I'm going to state the obvious, but it's as good stewards, we should be planting trees today for folks 20 years from now to be staying in the shade of, which happened in the late 80s and 90s with your work. The implications of what's happening in DC today, we feel it today. But I'd like you to talk a little bit about looking forward to what won't be done 20 or 30 years from now, because we're not making those foundational investments today.
So I want to make sure I love the question. I just want to make sure you mean what's not happening because of the destructive forces, or in general, what should be the strategy of what we're doing today? Yeah, I mean, your underlying question is exactly right. And again, the health and well-being of this entire ecosystem, of which all of us are in some different part of, really requires the continual investment in overwhelmingly basic, fundamental research, and also solving the problems that are not investable by investors. I mean, so anything that slows that down will have devastating consequences. Now, maybe not next year, but it's the year after, the year after, the year after. I mean, I am quite certain lurking in those 3 billion bases of the human genome are some functional elements that we can't even wrap our heads around. I don't need to remind people that when the Genome Project, we didn't know small RNAs existed, and we didn't know they did. There's things like that. We need incredibly basic, because some of those are going to have variants in them that are going to not only cause diseases that might be diagnostic opportunities, but might also give us insights about new therapeutic avenues. And so you're absolutely right. It's the stuff that it seems incredibly esoteric, and as I'm sure others know, let's just take anything like CRISPR or PCR or so many of these things. The fundamental discovery that led to them was, way off in some other direction at the time. And that's what has to continually be invested in. And some of the rhetoric that goes on about, doing too much, I'm not saying NIH is perfect, and it's not, but to destroy it or to do any significant damage is very scary. And it's also scary from the point of view of thinking of not only you said plant seeds of discovery for 10 years from now. Let's think about the workforce. We got to get the bright minds not scared of going into this. Yeah.
I noticed that, I appreciated your talk, it was very informative. I noticed you didn't really touch much on behavioral medicine.
Yeah.
And one of the questions I had, I've been waiting for it, and I know that there's stuff going on there, but to what extent do you think that this activity has affected or is affecting the actual definition of diagnostic categories in behavioral medicine?
Yeah, no, it's a great question. And there's so many different ways I can answer it. I didn't emphasize any disease area, to be fair. And part of the reason, the good news why I didn't have to is, and maybe I didn't say this strong enough, what has happened, especially over the last 15 years, is that all the other institutes at NIH have taken up genomics. When the Genome Project first ended, really, even when I took over the institute in 2009, the only institute doing significant genomics outside of ours was the Cancer Institute. All the other institutes weren't really doing it. One of the things I'm proud of is the NHGRI. I got all the institutes, including the National Institute of Mental Health, Child Health Institute. They started to say, those, like in behavioral areas, we're going to invest in genomics in our area. Or the Aging Institute invested big time in Alzheimer's and dementias. So there's a lot of excitement going on in those. But you said something at the end, which is spot on. And that is that what we believe this is going to be able to do, and actually there's some great data coming out in some of these areas, is to stratify patients. There's so much of a lumping. I mean, let's just take what's in the news, although don't listen to what's in the news coming out of the secretary's mouth with autism. But autism is a great example because it's not one disease. And it's an oversimplification to think of it as a one disease. And I think genomics, if nothing else, is going to teach us it's 10 diseases, and then we will be able to stratify and be much more powerful in our approaches. And you're going to see this almost in all the psychiatric disorders as well. But you're seeing it in cancer. I mean, and you're seeing across the board, all these things, these are all multiple diseases. Yeah. Okay, is it this one, or had you already promised it away?
The J and the Jimmy.
Okay, I'll try to answer quickly.
Dr. Green, that was terrific. My head is spinning. I'm trying to keep up with you. It's really, really wonderful. 2 questions. First is, as an individual, should we all be getting our genomic sequence?
Okay.
And have you gotten yours?
Yeah.
And then secondly, maybe as importantly, you're from St. Louis. Where'd you go to high school?
Oh, okay. LaDue Horton Watkins, that's the easy one, and you are right. That is the answer is my attitude, and I've lived by this, is that I don't think right now it's worthwhile to get your genome sequenced unless there's a health condition that you are concerned about that would tell you that would be informative. And so I can tell you without, so there, I did bump, I'm healthy, so don't worry about me, I'm healthy, but I did bump up against something based on a little bit of family history where it probably was a good idea to just rule out a few potential mutations, and so I did get my genome sequenced about 2 years ago. But again, I waited until I felt that it was necessary. That's what I'll say now, but we might be changing that, over the next 5 or 10 years. But that would be the answer I would give. Yeah.
Yeah, my question kind of follows up with his first question is, like, what are people doing with
Oh, I thought it was where did you go to high school, but go ahead. No, sorry. Yeah.
But I mean, like, what are people doing with this now? Because with the diagnostics, the question is always how does it change the course of care, right? So if you have, and you're going to get something, but there's no therapeutic product out there that's available.
So I mean, there's so many answers I can give. I mean, first, you go in, you don't always. I mean, there are cases you do have therapeutic options, and even some of the, there are ways of intervening to be able to do, even if it's the therapeutic. It doesn't. Sometimes it's a drug, sometimes it's other types of therapy. I will also tell you, and maybe I've been extremely moved by the following. We stood up something called the Undiagnosed Diseases Program, which eventually became an Undiagnosed Diseases Network. WashU is part of that, where people who have disorders, and a lot of times they just want an answer. They don't even know if it's genetic, and they just want an answer. And I'm telling you, if you talk to these families, when you could give them a diagnosis, that takes a huge burden off them, because sometimes they feel guilty, especially parents. They think it's something they did in raising the kid, et cetera, et cetera. So there is, and the other thing you do with a diagnosis is you may have an ultra rare disease, but what we have effectively seen as a community is it may be super rare, but you have suddenly given somebody a diagnosis, and now you can connect them to people around the world who have similar diagnoses. And as a patient group, they learn from each other, and in some cases, their aggregated information actually helps the clinicians taking care of this as a group, because they realize things that may have tried over here at work, maybe they tried on their patient, they otherwise would have never been part of that community, and that's very helpful to them as well.
Are they using it now? Is it reimbursed?
Well, reimbursement, I mean, my understanding is, and I don't know if there's any pediatricians in the room, they would be the best ones to know. And it depends on the state, depends on your insurance company, it depends on a lot of things. I think by and large for the evaluation of patients with suspected rare genetic disease, it's getting reimbursed. Okay, thanks so much. I'm here.